We have a 2 or 3 column strategy. We have very rational approach also to process development. This is not a scaled-up research method. It's not redundant. Every step serves a certain purpose. And still, when we now try to accommodate high-titer processes, we have certain limitations here around the protein A step, around intermediate purification, polishing, and I would, in the second part of my presentation, go through some of these limitations there and some possibilities how to address them. You see here, this is my first example. It's a study from Pfizer together with Cuno. You know, Cuno is sort of the standard in cell removal with lenticular filters. And what I liked about the study here is, apart from the fact that that it has addressed certain limitations in high-titer processes, that it really shows that lenticular filters, and I don't want to go into more detail here, are maybe good for much more than just cell removal. We can make them smarter, we can make them more intelligent. Maybe in 5 years from now this will also be validated as a virus clearance step, as soon as we can also integrity test that filters. And there's no reason why we shouldn't use a technology that is relatively cheap and relatively and not complex for more than what we are currently employing it for. Second area, and here I would like to make a little disclaimer because I will talk a little bit about membranes, but first of all I want to make sure that I don't get that I will not be misunderstood, because chromatography for me is a great tool. It is the working horse in chromatography. It has really led the way for also many years, and it will also in the future. So there's a clear reason for using chromatographic resins, and I don't think that we will ever get away from these standards. This is from another survey that I got across recently, and it surprised me a lot that apart from filtration, and again this is not a surprise, more than 50% of the respondents were of the opinion that chromatography can also be made single use. And this doesn't differentiate regarding commercial scale or clinical scale, but this was for me a striking result. So what are the options that we have available? This is resins, diffusion control. These are membranes, convection control. We have all sorts of intermediate versions in between, like monoliths, like perfusive particles. And this is the range that we can choose from today. And since we are able to do so, we have seen convective materials like membrane adsorbers really dominating some niche applications in chromatography, and it's again very interesting to see that they are growing, and that's this year's result again from Eric Langer's survey, that they are even outperforming disposable bioreactors, which everybody probably thinks is the hottest area at the moment. So for convective media, they work in polishing when it comes to flow-through applications, and they work in capturing when it comes to the purification of large molecules. Again, next door, factor 8, vaccine manufacturing, virus purification, and so on. It leads to the situation that today we are talking about a consensus sequence in antibody manufacturing where the capturing phase is dominated by resins because they offer the capacity, and the polishing phase is beginning to be dominated by convective material, because here we don't need high capacity, but we need throughput. And that works, and that's why I think the combination of the two is a very good idea. And it goes further because as a result of the possibilities that we have now, following the QBD initiative from the FDA and the design space that is open for development, we have even now recommendations how to replace resins with membranes in the polishing arena. And there's even more good news, because when it comes to new technology, everybody asks the question, first of all, who is using that? Second question is that single sourcing? No, it's not. And I don't want you to read this poster here, maybe just the headline. Companies are beginning to implement new technologies in their portfolio, and at the same time they are validating several or at least 2 vendors. And that's good news, because most of the players that are active in the resin area are also nowadays providing also convective material. So, changing gears, I'm going backwards in the process now to the capturing step. And this is an area where we talk about protein A a lot, and it won't hurt you too much, Günther, what I have to say here. Because some of the information has to be interpreted, first of all. Lonza, again, says, and this is Jim Davis, if we increase the titer from 1 to 10 grams per liter, then protein A cost, in relative terms, will dominate even further. Now, if that is still $20 per gram, who cares? So the overall cost is much more relevant than the cost distribution. But if we look deeper into this fact here, that protein A is some sort of a concern for many users, we see that many of the respondents from the Langer survey, they are actually considering alternatives. And the reason for that, and that's my personal interpretation, is it does not make any it does not cause any concern in the large scale, because you can dilute the cost over the 200 cycles that you run protein A with, but it does raise concern in the smaller scale, where you run 5 cycles and throw the material away, where you cannot dilute. And this, of course, is relevant for probably 80% of your development candidates, because that's the fraction that does not make it to the market. So, what are the alternatives here? First of all, alternatives to protein A use at all, replacement by other ligands, such as protein A, mimetics, mixed mode, cation exchange. Well, to add a grain of salt here, the issues that I see and that everybody else probably also sees are selectivity, scale-up, reproducibility, comparability when you change the technology later during the process. There may be other alternatives, such as precipitation of either the product or the impurity. Again, this is very promising, and this is from Duncan Lowe here, who actually, and that is more of a hypothesis, thinks that it can replace protein A and yet yields the same sort of quality of the end material. But I have worked in that area. I did precipitation of monoclonal antibodies, and it's not that easy. It's really, we have added polyethylene glycol up to 19%, and that is a lot of material. So it may work, and I think it makes absolutely sense to work into that direction, especially when it comes to the precipitation of impurities. I think that holds some promise, but other than that, it is still not there, and it's far from being an alternative. Protein A can also be used, of course, in an alternative format, with the goal to intensify the use by reducing the volume of the columns. And again, not going into any of the details here, I just wanted to mention that we talk about simulated moving bed, we talk about now expanded bed chromatography that is coming back, and the companies that are active here are almost too numerous to mention. And again, my assessment, you may not be able to read it, seen in that regard developments from Symbiosis in 2000 that ended up with actually not ready to use. Then we have seen some other developments with nanoparticles, where protein A is linked to a tobacco mosaic virus on the surface, And very recently, and I think that is a very promising approach here, protein A expression on the surface of polyester beads from a small New Zealand startup company. So there is some promising research in that area, and I think we should really closely watch what is going on. So I skip 3 slides here to come to my conclusion. What will the future of downstream development hold? I talked about cost and capacity issues and limitations. Well, I think there's 2 strategies to take that on. One is operational excellence, and we have to do that. We have to become more efficient. We have to run the same race faster, but at the same time we also have to watch another approach, and that is the innovation approach, to run a different race and make innovation not only on the product substance level available, but also on the technology level, because innovative products also require innovative technology. And in that regard, I think Asia plays a very, very important role. And I make a long story short here, because we have several cycles of migration of modern biotechnology into Europe, into Asia, and we are all expecting India and China to catch up now. And that's my conclusion. I pretty much hope, and I think we can also observe already, that this new wave will also lead to a new wave of adoption of innovation and new technology. We see that with disposable bioreactors. We see that with a lot of requests that we are getting here from companies in Asia that have no access to existing manufacturing capacity and that are considering starting from scratch and starting with the most recent standards and not with what they are supposed to use in the Western world. And I think that is very good news, and that makes me also very optimistic for the future of biomanufacturing here in Asia. So that concludes my talk. Thank you very much for your attention.
Thank you, Uwe, for that nice overview.